Thank you very much, Bruce, for the introduction. Black mussel cracker, or the punskop, is still one of my favorite fish as well. Always have a soft spot in my heart, but thank you very much for the introduction. Good evening, everyone. It's wonderful to be joining you, all of you online. I think this is the power of Zoom these days, is that we can connect all over Africa and all over the world, with some people joining from England. I actually took a gap year in Buckinghamshire as well, so I again have a very fun spot for that part of the world. But thank you all very much for joining. Tonight, I just want to talk a bit more about the platform that I currently work on as a researcher and that I manage, called the Acoustic Tracking Array Platform. And it's really grown from very humble beginnings to the network that it is at the moment. So there's always been an interest. People have always had an interest in studying the movements of animals. I think you see these animals going somewhere and you see birds arriving at different times of years and you think, geez, I wonder where those animals are going to. So obviously with this curiosity, the technology has developed and enabled people to be able to track animal movements all over the world, ranging from tiny insects such as butterflies and dragonflies, all the way to your large predators such as wolves, tigers, and leopards. And of course, the bird fraternity or community is very well versed in tagging birds, particularly using the rings as well. So this tracking technology has really blown the door wide open for tracking movements all over the globe. But what about animals that you don't see on land? What about the animals that live under the water that make it a little bit more tricky to track? So what I want you to do is hold your breath and let's take a dive under the water. I just want to touch on a couple of the ways that you can track animals. The first is dart tagging. Bruce and Gareth Jordan gave a really great talk about 2 months or so ago, a couple of weeks ago, on the Oceanographic Research Institute's cooperative fish tagging project, which involved these little colorful items called dart tags. And what it essentially does, you place it into the fish, as you can see on, this is a beautiful black mussel cracker. In this black mussel cracker, that little yellow strip that's sticking out, the little dart, which you can see in the top left corner, essentially hooks behind a bone in the near the back of the animal. It's sort of the base of the fin. And once you pull it out, it's tight in the fish and it's not going anywhere. But the program that Bruce was working with and still actively tags for provides phenomenal movement information. But the unfortunate downfall is that when you tag an animal, you have that location, but you need somebody else to first catch that fish before you know any kind of movement information related to that animal. So if an animal gets recaught, you've got a point of tagging and you've got a point of recapture, so you can then start piecing together where animals might have gone. But what about that missing period in between? What happens to them then? Do they stay in the same place? Do they go and come back? So this is where acoustic telemetry comes in, where you can really start filling in these missing gaps. Acoustic telemetry pretty much involves 2 pieces of equipment. The first is an acoustic receiver. The second is a transmitter or a tag. And you can think of the tag as a cell phone and the receiver as a cell phone tower. The cell phone can only work within the detection range of that tower. And this is really the gist of how telemetry works, where your transmitter sends out a sound signal, and if it is in the detection range of a receiver, that receiver can pick up that signal and store certain information. Now, what we do is we're very fortunate, it's hard work, but somebody's got to do it. We go out and we actually have to catch our animals. And a lot of the time it's using rod and line. So field work the research is hard work, but, we enjoy what we do. And a lot of the time you can either put tags on animals, such as your big sharks or turtles. But a lot of the time we actually put the transmitter into a fish via a small surgical procedure. And this little graph just shows that process taking place. So it's really just a small incision, you insert the tag, and then you stitch it up really nicely with 2 sutures. In smaller fish, we tend to anesthetize them just so that you can limit moving, and the utmost care is taken when handling these individuals, because obviously we are very invested in the well-being of these animals, because this telemetry game is very expensive, and we try and take care of, obviously we want the well-being of the animal, but we want that tag to be able to record or to send out as much information as possible. And then in larger sharks, what tends to happen is you can bring them alongside the boat, and once you turn them over, the same with big rays, they go into tonic immobility, which essentially prevents them from moving, and you're able to do the surgery quick and easy, turn them over, wait for them to be fully recovered, and send them on their way. Now, using the global norm of a size scale, which is a standard highlighter, we have a various range of sizes. All these transmitters come in multiple different sizes. From the right, you've got one that goes from 4 months and increases in age, and the longest one that you see, the biggest transmitter, can send out a signal for 10 years, which means you can actively track an animal for 10 years. This has really just exploded the field as well, because now we can track a full life history of an individual. So say an animal only lives for 10 years, or no, let's call it about 12 or 13 years, and it matures in 2 or 3 years. If you take that animal at maturity at 2 or 3 years, you can essentially get that entire adult life history phase. You can figure out where they're going, what they're doing, are they spawning, are they migrating? So it really answers a whole bunch of questions. So we've got our transmitters, what our receivers look like. We've got some beautiful photos which a colleague from ours from Ori took, Dr. Ryan Daly. And this is what a receiver deployed in an estuary looks like. This is the beautiful Mtentu estuary along the Wild Coast. And this is another receiver that's deployed in the Bushman's estuary. Now, a lot of time, what you can see on the right is a temperature logger. This logger records the water temperature every hour on the hour, and it helps us link the movements of an individual or of a species to the water temperature, because fish are ectotherms, which mean they are cold-blooded and they cannot maintain their body temperature. So water temperature is one of the major factors that drive their movements. So by linking this information together, or it's really important to record temperature so that you can make these links. When you look at the nearshore environment or the inshore environment, you can get receivers, or some of them are deployed or they are deployed at sea but are diver retrieved. This is generally in depths that are less than 30 meters within the safe diving levels for scuba divers. You can see the receiver at the top of that little loop. And for those that are deeper than 30 meters, we actually have them on release systems, where at the bottom part of this picture, that's the release mechanism. So you actually don't even need to get in the water. You just put a little transponder over the boat, you punch in a code, sends down a signal, and the whole unit pops back up to the surface. So this has really been quite handy when you've got external people that potentially help you maintain the network, which I'll touch on a little bit later. So in a nutshell, we've got fish swimming around with tags in them. We've got a whole bunch of receivers that we've put in whichever water body we want to study these animals in. So whether it's a riverine environment, an estuary, or even at sea. So you've got your receivers in the water, your tags send out their signal, and when in the detection range of a receiver, the receiver records the unique ID code of the fish, as well as the date and the time. So if you put all these pieces of this giant puzzle together, you can follow an animal or learn more about this animal's movements over a long period of time, or even a short period of time, but you know where that animal is a lot of the time, as opposed to dart tags where you've got the 2 points in time. I don't want to draw attention to any of the numbers on the page, but I just want to show that the curves all go up over time. And this is the number of studies using acoustic telemetry across the world over time. This field has completely exploded and is now one of the most popular methods to study the movements of aquatic animals throughout the world. This has essentially led to the development of large-scale networks of acoustic receivers across the globe. You've got some in the east coast of America, such as Rayon, the FACT network, ATAP, they tag like big, large animals. You've even got some huge extensive arrays throughout the Great Lakes that straddle America and Canada. You've got our partners down in Australia, the AMOS network, as well as the Europeans who have got their own network called the European Tracking Network. And here's us down here in the global south, the Acoustic Tracking Array Platform, and we are very much holding our own, albeit we are a smaller network. So who is to blame? For all this telemetry nonsense, who is to blame? This man right here, Professor Paul Cowley. And he's a very dear colleague of mine and a very good friend. And he got an active interest in this by attending some conferences in Europe. And he had a good friend, Dr. Tu Nasher, who you see pictured on the left. And between the two of them, they came up with, they were very fortuitously funding calls that came out between South Africa and Norway. And these came out way back in 2002. So this has been a 21-year collaboration, which has been absolutely phenomenal and really, really exceptional. Or it's really the grassroots of telemetry in South Africa. And as I mentioned, very humble beginnings. So this all started with Walter, the dusky cob. And he or she, we're always never quite sure of fish, was tagged in the West Clanemonde estuary down in the Eastern Cape, which is near Port Alfred. And this is where Bruce and Judy are. Shout out to Clanies. And the fish was tagged in the Clanemonde estuary and was actively tracked, which means people, you can see that picture down in the bottom right, they literally followed this fish with an active receiver, which is that little box you can see down in the bottom right. And they tracked this animal for a solid week. This is what the honors class, the ichthyology honors class at Rhodes University, this was their project. So this fell onto them. So they sort of tag teamed and followed this fish all over the place. But the main point is it was one fish, one species, one estuary. This eventually branched out into the Great Fish Estuary, which looked, there was a big project going on looking at the movements of spotted grunter and dusky cob, which are two important fishery species. But this then became many fish, two species, but still only a single estuary. Slowly but surely, more and more students came on board and more projects were added. And this sort of set out a whole bunch of receivers were deployed in multiple estuaries throughout the Eastern Cape. And this became many fish again, many species, and many estuaries. And all these projects were really successful. The initial projects were really focused on estuary-dependent species. And the reason is they all have a typical life history, and it makes them quite easy to study within a small area like an estuary. So what this means is that the adults generally spawn at sea or lay their eggs. The tiny little early juveniles, I mean small little guys, 1 to 6 centimeters at most, recruit back into the estuary where they live as juveniles for 1 to 3 years in these nursery environments. And as they get bigger, they go back to sea to spawn, and so the cycle continues. And the reason a lot of the estuary, the reason why the study was so important initially was because all the species that we studied, which include white steenbras, spotted grunter, dusky cob, and leofus or garrick, are all in a pretty bad condition, with a lot of the species being overexploited or completely collapsed. If you look at dusky cob, there's predicted to be between 2% to 5% of their spawning biomass left, which means if you've got 100 animals, 100 adults capable of spawning, only 2 to 5 fish are left. So making understanding the movements of these animals is even more important, especially when they are actively targeted by multiple fisheries. But what about animals once they leave the estuary? What then? So you've got these pinpricks in these estuaries, and you know what they're doing in these refined systems or these confined systems. But what happens when they leave? What about these coastal movements? This really led to this huge partnership, or this brilliant partnership with the Ocean Tracking Network, which is the Canadian-based global network, the Global Tracking Network. And Paul Cowley was in contact with Fred Worski, who is the executive director of Ocean Tracking Network. And between the two of them, OTN very kindly gave us a loan of 100 acoustic receivers, which started the backbone of the network that I will talk about shortly. But this really opened up the doorway to study so much more than just estuary movements of animals. We could now look at things like coastal movements, annual migrations, differences between different areas. Is one species tagged in Eastern Cape doing something quite different to the same species tagged in the Western Cape? So it really opened up this whole new world. And this led to the current network that we have at the moment. So as I mentioned, this is the Acoustic Tracking Array Platform. And we currently have approximately 250 receivers in the water at any given time. This number fluctuates depending on the projects that are on the go at the moment. For example, in the Cowie Estuary, we currently have clogged up the system with 25 receivers. Down in the Breda Estuary, a couple of years ago, we clogged up the receiver with 15 estuaries. So this number generally fluctuates, but 250 is more or less the general number. The array covers 2,200 kilometers of the coastline from False Bay all the way to Ponta de Ora, with a couple of receivers either end of the limit. The orange receivers on this map are representative of those deployed in estuaries or harbors, and all the white dots represent the receivers deployed in the nearshore environment. I just want to really acknowledge all our deployment collaborators. This is not a one-man show. We would not be able to do this without the help of these brilliant, brilliant people, from shark spotters all the way to Ori on the East Coast. It's really without their help, we wouldn't have the network. And all these people, all these researchers that are actively tracking animals or contributing towards this telemetry research, share all their data with us as the ATAP, and we consolidate everything into one central database. So it's really with teamwork really does make the dream work in this situation. So I just really want to acknowledge everybody that's involved. So what's in the water? What are we tagging? So obviously over time, the number of animals tagged increases, but this is what we expect. And at present, over 1,800 animals have been tagged across a multiple range of different species. This includes 15 fish species, 24 shark species, 9 ray species, and 3 turtle species. But overall, in terms of actual number of animals tagged, 46% of those are sharks. So they form the majority, followed closely by fish with 40%. At present, we monitor 43 different species and about 950 active individuals. So there are still animals swimming around with a tag in them, but the battery within that tag has now expired. So it's no longer emitting signals. So we unfortunately don't have information on those animals. But at the moment, 12 fish, 3 turtles, 9 rays, and 19 shark species are being detected. This includes awesome sharks. This is Team Ori on the KZN coast tagging this beautiful shark. We've got blue stingrays down in the Knysna estuary. There's this interesting aggregation. So all these rays just aggregate in the Knysna harbor. And we've managed to tag a couple of them and really interested to see what they're doing, because why are they here? Is it because they're spawning? I mean, pupping? Is it because they're mating? What is going on? 2 large adult dusky carp in the Breda estuary, arguably the last population stronghold for the species in the country. 2 aquarium rehabilitated turtles. So these are guys that are often washed up on the shore, either due to injury or being slightly ill. The brilliant turtle conservation teams, either at the aquariums or elsewhere, rehabilitate these little guys and send them on their merry way when they are fit and strong and healthy. And lately have been epoxying acoustic tags on them. So we're hopefully going to learn more about some turtles too. Right, on to some movements, some results. And this is always the exciting aspect of things. So I might ramp up my enthusiasm a little bit, but this is, I'm going to try to highlight various aspects of movements using different species as examples. So when we think about residency, we think home loving, they stay in one place, this is where they live. And dusky cob is really a good example of the species. For a couple of years now, we've been monitoring the movements of very large adult dusky cob. The biggest one that was tagged in the Breda estuary was 1.6 meters, which is phenomenally big. But the take-home message is really that they are so much more resident than previously thought. I mean, these are big animals capable of moving quite a far distances, but yet they stay in a small area. If this works, and I hope you can see it effectively, this is just a little animation showing the fish moving between the Breda estuary, which is on your left there. All the individuals or all the colored dots represent individuals that were tagged within the Breda estuary, and the black individuals are representative of those tagged in the De Hoop MPA, which is marked by the red boundary. And what's clearly evident is that the fish that are tagged in the De Hoop MPA largely remain within the De Hoop MPA. So this area is clearly very important for these large adults. The colored individuals, so the ones tagged in the Breda estuary, on the other hand, seem to move far, far more greater distances, yet still come back to the Breda estuary. So a very important area for the species, particularly those tagged within the MPA. This is a little blue stingray. These guys are known to be very resident. This is a species only found in South Africa, so it is endemic to our waters, and they don't like moving. They stay in a small area, and they sort of do inshore, offshore movements. So when they are inshore, they're coming inshore to pup, predominantly the females, and then they slowly shift off again, offshore again, when it's no longer pupping season. But the interesting component that we found in this study was that, yes, these animals are home loving and they like living in a small area, but man, oh man, are they the champs in terms of long distance movements when it comes to undulatory rays. So these are the rays that kind of frill, because these guys kind of swim with that fan motion within their wings, their fins, wings, and the furthest distance we recorded for one of these individuals moving was 450 kilometers, which is the furthest recorded movement for an undulatory ray in the world. So we, South Africa, are the home champs, our little endemic blue ray. What about mullet species? Everybody just looks at these guys and goes, bait fish, let's catch something bigger, like a leervis or dusky cob or, something else. But we look at these guys and go, this will be interesting to study the movements of. And the reason why is because we've looked at leervis and we've looked at dusky cob, which are two fish-eating species. So they are piscivorous fish species, and they actively take mullet. And this study is ongoing in the Cowie estuary. We've got 2 PhD students on this project. I think I saw Bantany's name pop up on the screen just now. Hey, Bantany. And despite they're so overlooked, everybody just go, mullet. But for starters, we were actively targeting 3 species, and we ended up unintentionally tagging another 2 because mullet are infamously difficult to tell apart in the field. We did pretty well, but we had to use genetics to sort of confirm the species. But even these small fish species are doing phenomenal things when it comes to movements. This is really preliminary stuff. We haven't downloaded all the receivers and we haven't got all the information yet, but just such interesting movements. And I'm still trying to decide whether these movements, you see a couple of dots right at the bottom of each graph, whether these are actual detections or not. But if it is, those fish are screaming down on the tide. So we're still going to find out more about this. Really interesting stuff coming out of this project. What about big sharks? You always think of ragged-tooth sharks or like a white shark as these solitary animals. They kind of do their own thing, and maybe interact sometimes, especially when it comes to mating areas or pupping areas or even just gestation areas. Raggies are tend to known to aggregate quite a lot on the East Coast, and I think it's linked to gestation. But what about ones tagged elsewhere? So there's a lot of animals that have been tagged in Algoa Bay, which was linked to work done by Dr. Malcolm Smale from Bayworld. He's now with Nelson Mandela University. And what we actually found was that these animals are pretty social. So all the blue dots that you are seeing in that figure is showing how these animals are interacting. And this is within a small area. This is Algoa Bay, which is near Port Elizabeth or Gqeberha. So it's a really small area, but a lot of socializing going on within the species, which is brilliant to see. Just out of interest, the yellow dots are representative of leofus, and the gray dots are representative of cob. So there is some interaction between them as well. What about looking at movements differing between males and females? Now, one aspect which I'm always very jealous, I personally am a fish researcher. Fish is what I love. I can appreciate sharks and rays, but fish is my passion. But one benefit that sharks and rays have over fish is that you can externally sex them. So by the presence of claspers, which are the male organs on sharks and rays. So you're able to further break down movements into male and female movements, which you can't do so easily with fish. This is also very preliminary work. This is a project ongoing with Shark Spotters and Two Oceans Aquarium, as well as us, and it's looking at spotted gully sharks and 2 or 3 other species within False Bay. And preliminary information's shown that a couple of sharks tagged at Helderberg MPA within the False Bay. The females do one thing and the males do the other thing. So females tend to go westwards and males tend to go eastwards. So we're not too sure what's happening there, but that was a really interesting preliminary result that we saw, or observation. So interested to see what else comes from this project. Then in terms of coastal migrations, this is a duck bull ray, these beautiful, beautiful animals, also known as bull ray. I think we knew that these animals were pretty far movers, but we didn't quite realize how capable they were of moving. These graphs are quite low quality, so I apologize if it's difficult to see. But on the left, we've got, this is the connectivity. So you can see an orange line connecting a couple of points. This is the extent of movement that an individual showed, or the species as a whole showed in summer. Look at their movements in winter. So much more connected. So these animals are making huge movements in winter, undertaking these massive coastal migrations up and down the coast. So it was really nice seeing this of a ray species as well. Interestingly, diamond rays seem to do exactly the same thing. Sticking with coastal migrations, I mentioned looking at or tracking the full adult life history of an individual earlier. So leofus, beautiful animal, always going to have a soft spot, seeing they were my PhD animal, but I looked at the little juveniles. These are the big boys. This is the big leagues. These are the guys that everybody wants to target. But we tagged an individual at the PE Harbour Wall back in January 2015. At tagging, it was 90 centimeters in total length. So quite a big fish. Definitely on the cusp of maturity, if not mature already. This is that individual that was recaptured by an angler. Should have put a trigger warning. It can seem, as a fish person, you might be, oh my word, this massive fish has been killed, and how dare they, and blood and everything. However, the interesting thing that came from this fish having been taken out the water is we got a tag back. And we're like, hey, we got this tag back. So when we look back and we saw the history of the animal, we saw these annual coastal migrations to KZN. Now, leofus have a very well-known annual migration where they go from the Eastern Cape and Western Cape up to KZN during winter, and they return back to where they came from. And this is exactly what we saw. I want to point out the absence of this movement between 2015 and 2016 and 2019 and 2021. The reason why we're not seeing dots at the top is because there were no receivers in the water, or if they were, the receiver batteries were dead, so they couldn't hear. They were not listening out for those tags at all. So we can pretty much confidently say that this individual made that movement 1, 2, 3, 4, 5, 6, 7, 8 times. So this individual right here had a chance to spawn 8 times within its life. Learfish are only meant to live, Bruce, correct me if I'm wrong, about 14 years, 10 to 14 years, 12 to 14, which means at tagging, it was probably about 3 years old, and it lived for another 11 years, where 8 of those years, it was able to spawn. So this fish was actually nearing the end of its life. So if you look at it that way, this fish has actually contributed to growing the population so much already that you can almost justify this animal coming out the water. I mean, obviously it's nice to stay in the water, but if the fisherman has a license, he's fully entitled to take the fish out the water. But still, how amazing. This is what the 10-year tags can tell you. Now, this is a really interesting information. So this is looking at tiger sharks, and a tiger shark, it was a female, was tagged at Reunion, which is, if you bear with me, laser pointer over here. This is Reunion, and it was tagged, I can't remember the exact dates, but it was never detected in Reunion. And the reason why they tagged a whole bunch of bull sharks and tiger sharks as at Reunion is that they had a spate of shark-human interactions, lots of fatalities, and the government was trying to figure out what was going on. They wanted to implement a massive cull program, and researchers were like, give us a chance to learn more. So they tagged a whole bunch of these animals, and this tiger shark they tagged was never detected at Reunion. Next thing, it popped up at Port St. John's along the Wild Coast. Next to be recorded at Ponta de Ora in southern Mozambique, and then it went dead again, and nobody could really understand what was happening, only to be recaptured by fishermen in Madagascar. So this animal is making these huge, huge coastal movements, which I think while everyone knows they are capable of doing, tiger sharks are generally sticking to the coastal areas. So this is huge. This is a huge movement for an individual. So why do we do all of this? Why, at the end of the day, is knowing where an animal moves so important? One, it's super interesting. You learn so much. I mean, the insights you get into information collected using acoustic telemetry is just completely mind-blowing. But the second is it's absolutely crucial for management. If you think of species that are resident all year round, they stay in one place, they're not going anywhere. If you want to implement something like a protected area, you just put it around that entire population and they are safe. A for away, you've met all the right targets, this animal will be saved. But what about those that maybe do something different between summer and winter? Learfish, for example, undertake these massive coastal annual migrations. You've got the sardines that are moving up, which bring a whole bunch of species with them, like bronze whaler sharks. So implementing something that a protected area that might work for a resident species is most certainly not going to work for a species that moves and covers the extensive distances. So you might have to look at something that looks at splitting a protected area and covering portions of these areas that they occupied during different times of the year. So what can you do to help? Is there something that you can do in order to help fish and contribute towards management? And something that you can do is practice catch and release fishing. Obviously, there needs to be some better handling skills involved, but these are all things that can be learned through practice and through some training programs. There are some really great videos out online at the moment, but if recreational anglers, whoever's out there fishing, had to release one extra fish, then one extra fish would be swimming around. So there is something that anybody can do, and that is practice catch and release. I came across this many, many years ago. This was from a Canadian colleague, and she gave this presentation on science communication, and I always loved it. So humans, which are all of us, are storytellers. We love telling stories. This has obviously been passed down generation to generation. You and me are obviously humans. Scientists are you and me. So now anybody can be a scientist with these amazing, incredible citizen science projects. So all of us can contribute in some way. So scientists are you and me. So if we think about just some basic maths over here, we're going to cross out some humans, we're going to cross out some you and me. Scientists are pretty much storytellers, and I hope you take that in the right sense, not like we're making up things and fabricating lies and stuff, but I just love telling stories, and I really hope that came across tonight, because it's just this work that you do and tracking these animals underwater and seeing where they go. It's really interesting, and to be able to share that with everyone is just such a pleasure. So with that, thank you very much for listening. If you ever want to reach out to me, I'm available anytime. Please feel free to contact us. I've got our ATAP email address as well as my personal email address. And please follow us on social media. And with that, thank you very much, Ron.
So I am going to open the screen for questions and answers. Please either raise your hands, jump up and down, or use the reaction tools so kindly provided by Zoom. Maybe I can kick off. Talking about the suturing of fish, what's the mortality rate? I mean, I take it not all of them survive the procedure.
So at the moment, like, we obviously take really utmost care for these individuals. What I touched on but didn't mention a price tag was that we're looking at in excess of 450 Canadian dollars per tag. So it's an expensive field we're in, but you know, when you get a little tag that's got a battery life of 10 years, you can understand it. What's interesting I can confirm with you that of all the animals that have been tagged, I can maybe point out 5 that have succumbed to the surgical procedure, but there were some signs that these individuals may not survive and probably shouldn't have been released in hindsight, but these are things you learn, and these are things that we had to overcome right at the beginning. So when you're sort of starting out, researchers were really doing the tagging themselves, and as we brought vets on board and got more training, get more familiar with the process involved, and you learn to take better care of the animals. We've now incorporated a little wound gel powder, which is basically like a little powder. You put it over the surgical incision, and when you make it wet, it turns blue, and it basically makes a giant plaster over that incision site, so it keeps out all the nasty things and really makes the survivability of those released animals so much better.
Well, that's remarkable. That's really cool. And I mean, well, it's remarkable that I wouldn't have guessed that only 5 individuals succumbed, but that's wonderful. Thank you. Anyone else? Always thank yous and interesting talks from Sandra Harding. Letitia saying thank you for a wonderful presentation. Francesca, let's see, Professor Bruton, yeah, you're clapping either. Oh, there we go.
Thank you, Taryn. That was absolutely fascinating. In what direction is the improvement of the technology of acoustic tags going? Presumably increasing the detection range is going to be something important, but what can we expect in the future?
So there's a couple of developments in the pipeline. The company that we use with the tags is a Canadian-based company called Innovacy, and they are really, they've been kind of taken over by a huge aquaculture company. So they've sort of merged together with this aquaculture company. So their research and innovation team is massive, and they are making progress in this field all the time, largely driven through the demands of the researchers. So they're coming out with a receiver that has an increased detection range. So we'll be able to record animals that are moving further away from the receivers, because obviously now we are quite limited with the detection range. In estuaries, it ranges from about 100 meters to 600 meters, where offshore it can go to 1 kilometer, which sounds like a lot, but in a great big ocean, it certainly doesn't account for much. So the fact that we record as much information as we do is pretty astounding. What they're also coming out with is sensor tags. So they are tags that actually have little temperature and depth sensors on them. So every time it swims past a receiver, it records the date, the time, the ID, as well as a temperature and a depth that that individual is swimming at. So this starts bringing in like your what's the word I'm looking for? So sorry, I'm thinking now. So how deep they swim. So during certain times of day, you might have them swimming deeper, at night shallower. So you're learning more insights into animals like that. They obviously are come with a little bit more of a higher price tag. You get tags that enable you to figure out the tilt of an animal. So whether it is feeding, whether it's doing something else. And there are really exciting tags called predation tags, where you can tag one animal, and when it gets eaten by another animal, it's got a little sensor on it that gets eaten away by the stomach acid of the predator, and through that changes the transmitter's ID to send out a different signal. So then you've got a predator that's swimming around with a different tag, and then you realize, oh my word, it's been eaten. So they've got some really cool developments going on at the moment, and I can only imagine that's going to expand even further.
That's bloody brilliant. Cool. Jane, let's ask Jane, and then I'll move to the chat.
Hi. Thank you so much for this talk. Really amazing. So I'm just interested from the people side, in a sense, of how much buy-in you're getting from fisher people into your program, because suddenly it's like, oh, maybe I can fish again because you're going to put them back in the water and do something useful. So I'm just interested to hear a little bit more about that side. Thanks.
So it's a bit of a double-edged sword here. You've got some people that know how expensive the tags are. We tend to wrap a little reward sticker around them to try and encourage return of the tags, because if it's a 10-year tag and we get it back within 1 or 2 years, we can reuse that tag. And there's some people that now know how expensive they are, as do all of you, and they unfortunately hold tags hostage, which is an unfortunate turn of events, but it is what it is. You get other anglers that have really good buy-in in terms, and it's something we're trying to focus on moving forwards. We're wanting to start a project on a really nice fishery species called yellowtail. They are quite readily, they're on the green list for the SASSI eating list, so they're in a good place, the stock's in a good position, they are generally caught sustainably, but there's not much known about them at all. All the information we know about them comes from Australia, so we're hoping to start a tagging project soon on them. And what we're trying to do is get buy-in from the anglers that are involved in catching the yellowtail themselves. So you're getting some sort of sense of ownership of these animals, and you're wanting them to be an active part of the research program, because through that you can engage, you can encourage these fishers or anglers, whoever they may be, to pass on this sense of ownership to other people who may catch these individuals. So juggling the angler perspective is always very interesting, but generally the anglers are very supportive of the research that we do, and it's going to be more important moving forward. We've just come through a project linked up with the Norwegians, and the project focused on the 20 years of fish research that we've done, but we paired it up with social and economic aspects of the study. So that really rounded up what we did in a beautiful way, and it's only going to become more important in the future, and is something that certainly shouldn't be looked at the back end. You should approach it from the front. So definitely moving towards that way in the future.
Wonderful. Thank you very much, Jane, for the question, and Taryn for the answer. Let's Peter, your hand was up, but I see it's not anymore. If you do have do you still have a question, Peter? Let's unmute. There we
Unmute.
go.
No, thanks, Taryn, for an excellent presentation. And my question is in essence, in South Africa, with our current regulators and our environmental management systems, are they taking heed of the research that's taking place in the private sector, like yours, for example? And are they looking at, I mean, you were mentioning the dusty cob being threatened to the extent that it is, are they going to one day maybe close seasons for some of these species? And at what intervals do you think they would be changing the spawning seasons and opening and closing fishing periods as required?
Yeah, thanks, Peter. Nice question. So all the information that we collect and all the work that we publish and is available, we always make the managing authorities, which in terms of fisheries is the Department of Forestry, Fisheries and the Environment, we always make them very aware of the research that we're going and make the research, the results known. The recent example, or the dusky cob, is actually been active uptake by Cape Nature of this research. They are very interested in trying to adapt the current regulations for the species on the Breda estuary, because I know there's already the night ban in place. So through this information, we could probably look at tweaking regulations, but again, you've got to be very careful because anglers do like fishing and you can't stop that immediately. It's not fair on anybody. So I would imagine, we know that you get Cobtober, which is down in the Breda, and if you had to close any specific area or even a month, that would be, in my opinion, a month worth closing, just because that's when they are at their sort of thickest within the estuary. So you'd really have to work together with the communities, the small scale guys, the recreational guys, even the, what are they called? The fishing guides on the system. You'd have to work together to kind of come to some kind of agreement, because nobody ever listens too much to regulations, which is going, you're not allowed. So yeah, it's working together. But at the end of the day, we can unfortunately only provide them with the information. The uptake is up to them, and we can just keep pushing and saying, this is a good idea, you should really do this. But whether people listen at the end of the day, we're never really sure.
Lovely. Wonderful. Thank you very much, Peter. Liesel van As.
Thank you for the presentation. Two or 3 months ago, there was a presentation on unlocking, on sharing the ocean. And my question then was, because being situated in the middle of the country, you have a different feeling for the ocean. If you're talking about anglers, what effort is being made for the inland people when they're going down to the coast to make them more sensitive about the tagging? I assume every time that you talk about the tagging and the anglers, it's the guys that you see every day on the rocks and all around the coastline. But what about the generation that goes down for that 2 weeks holiday? What's the effort to approach the inland people to make them more sensitive?
It's actually a really good point. Thanks, Liesel. Because a lot of the time, I mean, you just have to think about Easter and Christmas, and I think we all know exactly what you're talking about. So this would definitely be a time to sort of target these areas. It's always difficult because as a member of the public, unless you're actively interested in some kind of environmental component, will you listen to a presentation or a public talk on your holiday instead of, you know, being on the beach or something? So it's quite a tricky thing to target. The inland, and I'm completely stereotyping, so please, I don't mean any offense if anybody says I'm completely stereotyping. Inland is largely focused towards carp and barbel and species like that, and it's generally, the freshwater fish are generally well released relative to the marine counterparts, because I think people think that the sea is a place of plenty and the fish will never run out. So you bring up a really interesting point. My mind's just ticking now about how to engage more with people that do their coastal or their annual migration down to the coast. So if you have ideas, I'd love to be in contact more, but thank you. I'm really got me thinking now.
Wonderful, Darren. Thank you. Prof. van As, I did, if you want to comment.
Yes, quickly, yeah. Sorry, for the catch and release being in inland, we have a fight with people and keep on releasing carp back into the system, and we always have a fight with people telling them carp is not our national fish, it's actually so no, I don't have ideas for you, but maybe you must hook up with the fishing organizations inland and the guys that does competitions inland, and obviously there you have people that would go out. So good luck.
Oh, thank you.
Wonderful. Just on that note, I'm going to ask Judy. Judy, you shared 2 links with us. Would you like to elaborate on that?
Thank you so much. Lovely, lovely talk, Taryn. Brilliant science communication. Really cool. I've just shared two links. One is for the Sambra website. We've got a section there for anglers, and we've got a lot of resources for anglers to have a look at. So anglers from around the country, not just coastal anglers, can have a look at those resources. Some of the links we're just working on upgrading our website, so that's the one. And then the other one is the Ori Tag website. And both of those have got videos on how to handle fish. They've got information on zonation of marine protected areas. So there's a lot of information about fish and fishing on both of those sites. So yeah, I just popped them in the chat just in case anyone's interested. Thanks, Rowan.
Thank you, Judy. Lovely. Cool. Then I have a question here that Moret sent me. Oh goodness, now I don't know what the question is. Oh, wonderful. Can this technology be used for other animals such as pythons?
Okay, so I think the one thing that I forgot to mention was that acoustic telemetry is limited to underwater environments, like satellite tagging, we can't use at all. The signal does just not break the surface of the water. So in terms of pythons, you can definitely use something called radio telemetry, which is what you'd use. It's essentially exactly the same thing, but just the land version. You can actually radio track freshwater fish. So you can use the same technology, just radio tracking doesn't work in saltwater, so you can't use it in estuaries or at sea. So in terms of pythons, you could definitely use something called radio telemetry and do the same thing, but you could also probably put a satellite tag or collar or something on a python and see where it ends up. That would be amazing.
Oh, lovely. And then another question from the chat from Lorette. Can you deploy these receivers in flooding rivers or for large rivers with seasonal change?
Okay, it's an interesting question. We actually experienced this in the October 2013 floods. No, October 2012, the floods that came 10 years ago. There was extensive flooding down our part of the world, Eastern Cape, and multiple rivers completely flooded. The banks of closed estuaries broke open. So it was quite chaotic. I was actually down in the Gouker estuary, down in Stilbaai in the Western Cape, attempting to start my PhD when we realized that the river was flooding because we saw a tree floating down. So we then realized this was useless and we abandoned mission, only to turn out that this was like the 1 in 100-year flood kind of thing. And interestingly, all our estuary receivers survived. They did not move. We tend to moor them in these big cement blocks that are about 45 kilos within the estuaries, and they didn't move at all. So it was really reassuring to know that if we put them in these systems, that it will be fine. The only thing so that was like a completely, random event. It's not going to happen again anytime soon. The only problem with very fast flowing water, is that the signal of the receivers gets really, really, really small, because with rushing water, you've got a lot of noise and you've got a lot of feedback. So the receivers tend to be quite limited in terms of their listening power. So while you could deploy them in flooding rivers, especially with rivers with seasonal changes, the detection range of that receiver, which allows you to listen for the tags, will be that much smaller.
Wonderful, 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 wonderful answer. Thank you very much. Are there any other questions from the audience?
Great talk. Thanks, Taryn. I really enjoyed it. Please, won't you tell us the marvelous story about the giant kingfish? For me, it's like one of the absolute stunning examples of acoustic telemetry that has shown us all something that we never knew about this iconic species. Thanks.
Thanks, Bruce. So I was quite torn between whether to include the GTs or not. Bruce and myself were working with a master's student who was really, really great. And he was focusing on the movements of giant kingfish or giant trevally that had been tagged along the South African coast, along the east coast, as well as at the Pinnacles Reef, which is a really famous area in southern Mozambique near Ponta de Ora, and which is like right at the border of South Africa and Mozambique. And what he found was that animals that are tagged within South Africa tend to go up and visit the pinnacles at certain times of the year, which tended to be early January, February. This is where the massive spawning aggregation, we think it's spawning anyway, takes place. And this draws fish from all over the place and has actually been recorded as the largest aggregation of giant kingfish in the entire world. This is quite a widespread species. They're found in the US, in Australia, down by us. And our species or our group of GTs do this movement, but it was only the South African fish where the Mozambican ones tended to stay in the area. They might have gone a little bit north, but they tended to stick around this Pinnacles Reef. What the South African ones also did, we also tagged a couple in the Mthentu estuary. I can't remember, I think it's Blue Planet, where David Attenborough focused on the giant trevally that came into the Mthentu estuary, and they sort of did this fairy ring motion where it was like a daisy chain motion, and they just kind of mill about in a figure of 8 or around in a circle. And it wasn't really understood what they were doing, and we still don't really understand the full extent of it, but we managed to tag some fish within the Mthentu estuary and within this daisy chain linking group of them, and their movements within the estuary were largely linked and largely driven by temperature. With cold spells, they would sort of go into areas that were more thermally stable, because they are generally prefer warmer water, a more sort of subtropical species. So their movements were really driven by these temperature fluctuations within the estuary as well. And there was going to be something else that I was going to tell you about them, and it's completely slipped my mind. So Bruce, if I've forgotten anything, please jump in. But it was a really interesting story, and it was something that we didn't expect. So sorry. In the rest of the world, in Hawaii and Australia, they are really limited to reef areas and coral reef areas. So our GTs are the ones that are moving the furthest in the world as well, which is something that elsewhere in the world they don't do. So ours are behaving really differently compared to everybody else. So it was just really interesting.
Oh, that's a nice story. Like a story. Wonderful, cool. Bruce gives it a big, massive thumbs up. And I think if there aren't any other questions from the audience, I'm going to call it a night on that wonderful story of yours. And thank you again, Taryn, for a wonderful presentation and entertaining us on this Thursday evening. My week's gone haywire. It's absolutely wonderful to have you as part of the family, the Unlocking Nature family, and all the best for the future and the research. And we'll have you back definitely to tell us some more. It's been wonderful, and thank you to every single one of the audience members who joined us tonight. It's been lovely to have you as well, to have some of you back, and to have new family members to join us. So on that note, have a wonderful evening, wonderful afternoon, or a morning, wherever you are in the world. Thank you very much for joining us. It's been wonderful. Good evening.
Thank you very much, Ruan.